Good morning, everyone. Good morning, Sunita. Good morning. Okay, what about others? Good morning, Priya. Good morning. Good morning, Brunalini. Good morning. Think still people are sleeping. Thank you, Sadhguri. Good morning. Okay, let's start. So, friends, in the last class, we have completed the prokaryotic transcription also, and we completed the eukaryotic transcription also. Good morning, Gautam. Now, friends, what we are reading now, we are reading post-transcriptional processes. Please continue, everyone. So, friends, if you very quickly recall, in case of a eukaryotic organism, whatever the mRNA that we synthesized, that mRNA is not biologically active, is it? That mRNA is not functionally active. Good morning, all. Good morning. To make the mRNA functionally active, to make the mRNA biologically active, we have to do certain processes. What do you call it, that particular processes, friends? Post-transcriptional processes. And friends, why do you call it as a post-transcriptional processes? Because friends, these processes are happening after the transcription. That's why, friends, we will be calling it as the post-transcriptional processes. Why we are doing this particular post-transcriptional process, friends? To make the RNA to become functionally active, to make the RNA biologically active. Good morning, all. Good morning. Please continue. So, friends, listen to me for the period of 20 minutes continuously. Later, I will solve all the doubts. Okay, in this particular processes of post-transcriptional post-transcriptions, what we have to do? This we can also call it as a post-transcriptional processes, friends. Widely we can call it RNA processing, which means we are modifying the RNA. Good morning, good morning. We are modifying the RNA. Friends, why we are modifying the RNA, friends? We are modifying the RNA to make the RNA to become biologically active, to make the RNA to become functionally active. Friends, all of you please kindly look into the diagram very carefully. This is the DNA. So here what you can observe, friends, you can observe the exons also, friends, and friends, even you can observe the introns also. And there is a very important site you people has to remember. We can call it polyacid. Friends, why we have given the name polyacid? Because at this particular polyacid only we will cut this site. All of you pay attention. We will cut this particular site and we will add the adenine. That's why, friends, we will be calling it as the polyacid. Please kindly don't forget this point. And friends, here comes another interesting point. What is that interesting point? When compared to the exons, introns are always larger. I mean to say, friends, when compared to the coding parts, that is exons, non-coding parts, that is introns, are very larger all the time. Please kindly do not forget the statement at all. And friends, listen to me very carefully, friends. So this DNA is going to give rise to what, friends? RNA. So the concept is very simple. Whatever the information that is present in the DNA, the same information, it comes to the RNA also. That's a common point. Once the information comes to the RNA, exons also comes here, friends, and introns also come here. Anyway, friends, we're gonna read that. Listen to me 20 minutes carefully. We will cut this particular polyacid and we will add this particular adenines. All of you pay attention. at the 5 prime direction of the mRNA. At which direction, friends? 5 prime direction of the mRNA, we are going to add an important cap. What do you call it, this particular cap, friends? We will be calling this cap as the methyl 7 guanine cap. What was that, friends? Methyl 7 guanine cap. I will tell you why we are calling it as the methyl 7. It's very simple. Anyway, we will read it. The guanine at the 7th position, it got added by the methyl group. That's why we can call it methyl 7 guanine. So where we are adding this, friends, we are adding this at the 5 prime group. And friends, there will be an addition of the tail. We can call it, friends, polyatyl. All of you listen my words very carefully, friends. Why I am using a word polyatyl? Because, friends, we are not adding 5 or 10 adenines. Friends, we will add nearly 200 to 250 adenines. That is the reason, friends, we always use the word polyadenylation. So capping we have done, polyadenylation we have done. Then, friends, finally what we are going to do, because this particular introns are not at all coding, they do not have any importance. So that's why, friends, we are going to remove this particular introns and we are going to join this particular exons. That's what we can call the splicing. So, friends, 5 prime capping, 3 prime polyadenylation and splicing, all together we will be calling it as what, friends? Post transcriptional processes. Please kindly don't forget that. And friends, I am just reminding to all of you, the RNA processing or this post transcriptional processes is very, very, very important for the 4 marker, especially in the part C. Friends, there are more than 7 to 8 times in one or other way we use it to get the question from this particular post transcriptional processes. Hence I am requesting all of you, friends, not to miss this under any circumstances. It is a very important, you have to know this. Now, friends, I listen to me 20 minutes carefully. I will explain very briefly. You will get a clear cut idea. You know what is this, friends? RNA polymerase enzyme. We already see that. RNA polymerase enzyme having what domain? C-terminal domain. We also know very well the C-terminal domain of the RNA polymerase enzyme undergo the phosphorylation. Friends, all of you people, please try to understand very clearly here which is coming out, my friends, in the green color. In the green color, whichever is coming out is the mRNA. Friends, whenever this particular mRNA coming out, immediately who will go and associated with that particular mRNA, friends? Capping complex will go and associate. Friends, the moment capping complex will go and associated with the mRNA, immediately what is happening? All of you pay attention. Immediately, friends, there will be a addition of the methyl 7 guanine. Friends, don't get confused. The moment capping will associate, listen my statements very carefully. Capping will helpful. Capping will helpful in what? Friends, capping will be helpful in the recruitment of the splicing machinery. Please kindly don't forget the statement. What, my friends? Capping will be involved in the recruitment of the splicing machinery. Once the splicing is completed, good morning Sumaya, continue. Once the splicing is finished, splicing machinery will helpful in the recruitment of the 3 prime polyadenylation machinery. My friends, this is how capping, polyadenylation as well as splicing will occur step by step during the post-transcriptional processes. Now, friends, from this particular slide, I want every student to pay utmost attention. Listen to me for the period of 10 minutes. I will finish the capping first, then I will solve all your doubts. It is really confused. It is really confused if you don't understand. But if you understand, it is very, very easy. And as I told you, it is most important for the part C questions. So hence we should not neglect even a single point also. So pay utmost attention. before understanding the 5 prime capping, first we have to know why we have to do the 5 prime capping. So friends, always listen everyone. This is mRNA, right? 5 prime to 3 prime. So friends, whenever we don't add the cap, the 5 prime end of the mRNA is not protected. What my friends, the 5 prime end of the mRNA is not protected. Since it is not protected, many kind of a vulnerable enzymes, what that vulnerable enzymes will do, friends, they will start degrading this particular mRNA. So friends, so the first and the most important point you people has to remember, to prevent the degradation of mRNA. What friends? To prevent the degradation of mRNA, we have to add the capping. And second and most important point, in the previous slide I have explained it to you friends, 5 prime capping will helpful in the recruitment of what kind of machinery? 5 prime capping will be helpful in the recruitment of the splicing machinery. Don't forget that. 5 prime capping helpful in the recruitment of splicing machinery. And here comes utmost important point. You will read in the eukaryotic translation. Friends, 5 prime capping, it is exclusively responsible for eukaryotic translation. How exactly 5 prime capping plays a key role in the eukaryotic translation? Friends, we will read in the eukaryotes. While reading the translation, you will get a clear-cut idea. And the last one, suppose example you remember, 5 prime capping is over, 3 prime polyadenylation is also over, splicing is also over. Then friends, the mRNA through the nuclear pore, where the mRNA enter into the cytosol, because cytosol is the area where the translation will happening, protein synthesis will happening. The question is very clear, my friends. How does mRNA entered from the nucleus into the cytosol? How does this particular transport can be guided? How does this particular transport is mediated? So that can be done by, of course, 5 prime capping. So 5 prime capping mediate the RNA transport. Please kindly do not forget the statement. It is what it is doing, friends. It is mediating the RNA transport. So these are all the major functions I want you to remember. Now look into the process very, very carefully. Don't confuse. Listen to me 10 minutes very carefully. Friends, this is the mRNA. Just listen to me without deviating from the lesson. This is the first nucleotide. This is the second nucleotide, third nucleotide, and so on. My friends, everyone pay attention. The first nucleotide of the mRNA, this is the first nucleotide having sugar base phosphate. So friends, in the first nucleotide of mRNA, at the 5th carbon, how many phosphate groups are present, friends? Of course, at the 5th carbon, there is a presence of 3 phosphate groups. Very simple point it is. Now friends, what we are adding to this particular first nucleotide of mRNA, we are adding the guanine. So friends, guanine also contains sugar, a base, then followed by the phosphate. And friends, guanine also at the 5th position, how many phosphate groups does it contain? Undoubtedly, my friends, it also contains the 3 phosphate groups. There is no doubt about it. Don't confuse, my friends. At the 5th carbon of the guanine, 3 phosphates are present. At the 5th carbon of the mRNA first nucleotide also 3 phosphates are present. 3 plus 3, 6 phosphates. But my friends, we don't add the 6 phosphates at all. We always add only 3 phosphates. You will get to know. Just listen to me carefully. We will add only 3 phosphates. We will add the 3 phosphate. We will remove the 3 phosphate. That's why, friends, we can call this particular linkage as the 5 prime triphosphate bridge. What is that, my friends? 5 prime triphosphate bridge. Kindly don't forget this. Now I will explain, just listen to me carefully. Same, this is the first nucleotide of mRNA, second nucleotide of mRNA, third nucleotide, fourth nucleotide, and so on. And this is what, my friends, guanine. Correct na? So you only look into this, friends. Once again, the mRNA first nucleotide having how many phosphates, friends? Having 3 phosphates. Guanine at the 5th carbon, it is also having 3 phosphates. Out of 6, we will add only 3 phosphates. And all the students pay attention towards this particular point. I have already told you, this is the guanine, right? Guanine at the 7th position. What, friends? Guanine at the 7th position, it is added by the methyl group. That's why we can call it as the methyl 7 guanine. Later we will read what kind of enzymes involved. Good morning, Saima. Please continue. Now, friends, all the students, please try to understand one very important point what I am saying here. You know, friends, guanine added by the methyl group, right? You know, this is called as a cap 0. What do you call, my friends? Cap 0. Now, this is the first nucleotide of mRNA, friends. This is the second nucleotide of mRNA. Friends, the first nucleotide of the mRNA, you can see, right? The second hydroxyl group of the mRNA also get methylated. You call it this, my friends, as the cap 1. And the second nucleotide of mRNA also contains sugar. That sugar at the second hydroxyl group, it is also got methylated, friends. This is what do you call, friends? Cap 2. So this is called cap 0, cap 1 and cap 2, so on. My friends, you can ask me, sir, is there any necessity of cap 0, cap 1, cap 2? I can say to all of you, cap 0 is compulsory, it's mandatory. But friends, cap 1 and cap 2, even though they are present or they are absent, it doesn't make any sense. What I want to convey here is that to the first nucleotide of mRNA, what is adding, friends? Guanine is getting added. Correct na? So guanine get added by the methyl group at the 7th position. Same, to prevent the mRNA from the degradation majorly. So that is what we can call cap 0. So my friends, cap 0 is compulsory. So friends, if you understand this particular slide, the entire capping is crystal clear to all of you. So first of all, I want every one of you, friends, to understand what kind of enzymes are involved. Listen, look into this particular slide very clearly. The concept will be very clear. How many types of enzymes are involved in this particular 5' capping? Friends, there are majorly 3 enzymes are there. First enzyme, friends, we will be calling it as, everyone pay attention, we will be calling it as what, friends? Phosphohydrolase. And friends, you know, this particular enzyme very widely called as triphosphatase. Instead of phosphohydrolase, we will be calling it as the triphosphatase. Friends, what do you mean by the word triphosphatase? Triphosphatase means, friends, which is involved in the removing of the 3 phosphate group. That is what exactly we call, friends, triphosphatase. And the second enzyme, my friends, we will be calling it as guanylyl transferase. I want you people to remember every single enzyme, friends. Guanylyl transferase. And the third one, friends, we will be calling it as the methyl transferase. One we called it as the guanylyl transferase. Another one, friends, we will be calling it as the methyl transferase. All of you pay attention, friends. This is the mRNA. Correct na? Friends, this particular mRNA, all of you listen. If you look into here, the concept is clear. Don't get confused. The mRNA having at the first nucleotide 5th carbon, mRNA having 3 phosphates. Alpha phosphate, beta phosphate, gamma phosphate. And what we have to add? We have to add the ZTP. So ZTP also having how many phosphates, friends? Alpha phosphate, beta phosphate, then friends, followed by the gamma phosphate. We all people commonly know very well. Now all the students pay attention, friends. What is the enzyme involved here? Phosphohydrolase enzyme or triphosphatase enzyme. How many phosphates it is going to remove? 3 phosphates it is going to remove. Listen to me. This triphosphatase enzyme from the mRNA, it is going to cut which phosphate, friends. You have to remember which phosphate is getting cleaved. It will cleave the gamma phosphate. When it cleaves the gamma phosphate, it's only one phosphate released in the form of a pi. And my friends, here what are the two phosphates that are left with? Left with is the beta and alpha. The same phosphohydrolase enzyme, my friends, from the GTP, it is going to remove two phosphate groups at a time. What kind of phosphate groups it is getting removed, friends? Beta and gamma. Beta and gamma are the two important phosphates that are removed from this particular ZTP actually. So here two phosphates are released in the form of PPI. Once again I am repeating, all of you listen. Phosphohydrolase or triphosphatase enzyme will remove the gamma phosphate from the mRNA. By that there is a release of pi. Very simple point it is. Next one, the same height phosphohydrolase enzyme from the guanine, it remove the beta and gamma. So my friends, all of you listen. What are the phosphates left right now? In the gamma, only alpha phosphate is left. In the mRNA, like this you have to read, beta and alpha will be left. My friends, listen everyone. Now this particular guanine with the alpha phosphate will come and associated with the beta and alpha present on the mRNA. Now it will become alpha, beta, alpha. Now the guanine with the alpha phosphate, who will take this guanine alpha phosphate and add it to the mRNA? Friends, this can be mediated by the guanylyl transferase. Guanylyl transferase is the enzyme which basically take the guanine alpha phosphate added to the mRNA. Finally, we added the guanine. There is no doubt about it. But I told to all of you very clearly, my friends, this guanine at the 7th position, what we are doing, friends, we are adding the methyl group. So friends, the question is very clear. Who is going to add the methyl group? 7-methyl transferase enzyme. Don't get confused. 7-methyl transferase enzyme, friends, it will just add the methyl group, that's all. But my friends, someone has to donate the methyl group. That is very important point, friends. Now, who is going to donate the methyl group? Friends, methyl group is donated by adenosylmethionine, which we shortly call it as ADOMET. Please kindly don't forget this. Adenosylmethionine or ADOMET donate the particular methyl group. Upon donating the methyl group, adenosylmethionine can be converted into homocysteine. Once again listen, upon donation of the methyl group, adenosylmethionine converted into homocysteine, that methyl group added here. That's the simple concept. Now all of you listen to me very carefully, friends. I told you now we added the methyl group to the guanine. I have told you even the first nucleotide of mRNA also get methylated. Second nucleotide of mRNA also get methylated, which I call cap1, cap2. See here, this is the first nucleotide of mRNA is getting methylated at the second prime hydroxyl group. That can be done by 2-O-methyl transferase. I don't want you to remember this because as I already told you, this particular process of cap1, cap2, it is not mandatory in the all cases. And quickly summarizing the capping, just pay attention all of you. So friends, the first enzyme called as phosphohydrolase or a triphosphatase enzyme, what it is doing, friends, it is it is removing one phosphate from the mRNA, which we can call gamma phosphate. Once it remove the gamma phosphate from the mRNA, friends, at the same time from the GTP, the same enzyme is going to remove the beta and gamma both phosphates. Then this particular guanylyl transferase enzyme will come and add this guanine alpha phosphate to the mRNA. Later, guanine 7-methyl transferase enzyme will come and add the methyl group to the 7th position of the guanine, and this particular methyl group is donated by the adenosyl methionine. Adenosyl methionine, upon donating the methyl group, it is going to be converted into homocysteine. Friends, this is what do you call 5' capping. Is it clear, friends? Now everyone please say yes or no. If anyone having any doubt, I will explain. And we will move to the 3' polyadenylation, everyone. Hope it's clear for everyone. If you have any doubts, you can ask me immediately. Okay, good, perfect. So friends, right now what we studied is what? 5' capping. Now friends, good, perfect. Now what we have to study is, friends, polyadenylation. All of you, great, perfect. Okay, so let me finish the polyadenylation. Later I will solve all your doubts. Whenever, friends, come on, be active. Whenever we are reading the 5' capping, we understood very clearly that 5' capping will do certain functions. Similarly, friends, polyadenylation also performs certain functions. What are the major functions performed by this particular polyadenylation? All the students, please kindly keep it in your mind, friends. This particular polyadenylation or the polyadenylation, all of you listen, it is extremely important, friends, for the stability of the mRNA. Very important point it is. You know, the mRNA can withstand for the longer time. What, friends? The mRNA can withstand for the longer time. It is just because of this particular polyadenylation only. Please kindly don't forget the point. And the second very important point, just quickly recall. I told you in the eukaryotic translation process, 5' capping plays a key role. How it plays a key role, we will read in the translation. Same way, however, 5' capping is required for the eukaryotic translation. Friends, even in the eukaryotic translation, 3' polyadenylation also very important. Just remember, polyadenylation is required for the eukaryotic translation. How it is required? When we are reading this process, you will automatically get it. And friends, why do I use the word polyadenylation? Because, friends, I am not adding one tail, because we are not adding one adenine, because we are adding nearly 250 molecules of the adenine. That's the reason, friends, we will be calling it as the polyadenylation. And what kind of enzymes that are involved here, every one of you pay utmost attention, friends. Look into the picture very carefully. And this is the mRNA. All of you listen to me carefully. I told you, friends, this is what site, friends? We called this particular site as the poly-A site. Why do you call it as the poly-A site, friends? Poly-A site means we are basically cleaving at that particular site. That is what exactly, friends, we will be calling it as the poly-A site. And we will cut that particular area and we will add the adenines at that particular area only. Don't forget the point. When you cut this particular adenines, now what is the part that we cut? Friends, of course, whatever the part that we cut, it immediately undergo the process of complete degradation. Kindly do not forget that particular statement at all. So cutting, polyadenylation, friends, followed by the degradation. Listen to me carefully. Don't get confused, friends. You have to know this. You can ask me, sir, is there any necessity to remember these sequences? You people no need to remember, but just understand this kind of sequences are present and what is their significance. This is the 5' end of the mRNA. This is the 3' end of the mRNA. Friends, in the middle, you can see it very clearly. What do you call it, this particular site, my friends? We will be calling this particular site as the poly-A site. Don't forget the point. At the 5' end of the mRNA, there is a sequence is located. We can call AAU AAA sequence. Please understand. And this is the poly A site. Anyway, friends, 100% we are going to cut this. No doubt about it. But just listen. 3' end, what kind of sequence is present? GU rich sequence is present. Now, you will get one doubt. Sir, does every single mRNA contain the similar sequences? I can use a word. What is the line I can use? I can use it is a generalized sequence present in most of the RNA. Listen to me, friends. I am using a word most of the RNA. I am not using the word all the RNA. In most of the RNAs, this particular AAU-AAA sequence is present at the 5' side and GU rich sequence is present at the 3' side, but not all the RNAs. I can use a word 90% of the RNAs. It is a most generalized sequence. Now, friends, just now we all people studied that there are only 3 enzymes are involved in the 5' capping. What are that particular 3 enzymes? We can call it phosphohydrolase, triphosphatase, guanylyl transferase, methyl transferase. These 3 are the 3 major enzymes. Friends, in case of this particular 3' polyadenylation, my friends, there are nearly 6 enzymes are located. 6 enzymes are present. One we call, friends, cleavage and polyadenylation specificity factor. And friends, I always encourage all of you to remember the short names only. CPSF, CSTF, cleavage factor 1. Actually, friends, there is a presence of cleavage factor 2 also. Then followed by poly-A polymerase PAP and PABP2, poly-A polymerase binding protein. I told you, right, we have to add up to 250 adenines. But anyway, friends, who will add this particular adenine molecules? Undoubtedly, polymerase is the enzyme which will add the adenine molecules. There is no doubt about it. But the question comes here, poly-A polymerase on its own, it cannot add 250 nucleotides continuously. My friends, this particular poly-A polymerase hardly will add 10 to 20 nucleotides. That's all, friends. Here comes an interesting point. So because it is having very low processivity, friends, whenever this particular poly-A polymerase enzyme associated with this particular poly-A binding protein, then what will happen? Friends, all of you listen to me very carefully. Then because of the help of this particular PABP2, this polymerase enzyme can add up to 250 adenines continuously. Anyway, let me explain you. Please kindly don't forget the sequences, I mean to say. At the 5' end AAU-AAA, 3' end GU rich sequences are present. So friends, all of you pay utmost attention here. Same, right? AAU-AAA and GU rich sequences, right? AAU-AAA, then followed by the GU rich sequences. Now all of you look into the diagram very carefully, friends, and here the pink color one you can see, right? That is CPSF proteins. The blue color one you can see, they are CSTF proteins. The green color one you can see, they are the cleavage factor 1 and cleavage factor 2. The light orange color protein you can see here, that is the poly-A polymerase, which is involved in the addition of the adenines. Friends, please try to understand, look into the picture very carefully. First, what happened, friends? This particular CPSF, what does this particular CPSF has done? This particular CPSF recognized the AAU-AAA sequences. Simple point. So friends, CPSF will go and associated with the AAU-AAA sequences. After that, CSTF, you can see, my friends, the blue color one, listen to me carefully. CSTF associated with the GU sequences. All of you listen to me carefully, friends. Whenever the blue color CSTF associated with the GU sequences, you know what it will do? It will bend the mRNA like this. See, mRNA is like this, friends. See here, mRNA is exactly like this. This part will be bent. Why? The part will be bent and it come near the poly A site. You can see, right, it bent and come near the poly A site. Then immediately who comes into the picture, friends? Cleavage factor 1 and cleavage factor 2. These two comes into the picture, friends. They will cut this particular poly A site. So once they cut the poly A site, immediately which enzyme comes into the picture? Poly A polymerase enzyme will come to the picture, and they start adding the adenines continuously. But I told to all of you, friends, it can't add more than 10 to 20 nucleotides. So that's why immediately there will be a recruitment of an important protein called as PABP2 protein. We call it as a poly A binding protein 2. That PABP2, friends, will come and start the process. What do you call it as, friends? Rapid polyadenylation, which means, friends, it is going to add nearly 200 adenine molecules. Please kindly don't forget the statement. Okay, now all of you listen to me once again very carefully. I'll quickly summarize, friends. First, CPSF will associated with the AAU-AAA sequence, CSTF associated with the GU sequence, and bend the mRNA near the poly A site. Immediately, friends, cleavage factor 1 and cleavage factor 2 will come over there. They will cut this particular poly A site. Then there is a recruitment of PAP. Then PAP start adding up to 10 to 20 nucleotide. Because of its low processivity, it cannot continued. Then PAP immediately recruit the PABP2. Then, friends, then there will be a rapid polyadenylation occur. So, friends, this process, what do you call it as, friends? This is the process we can call 3' polyadenylation. Very easy process. And what I want you people to remember majorly for the exam point of view, I want you people majorly remember the enzymes involved over there and their mechanism of action. So, friends, most of the time questions will appear from the 5' capping, and very important, where most of the time questions will appear is the splicing. And, friends, I can say in the process of splicing, hardly 1%, just 1% you have to remember, whereas, friends, 99% of the concept you have to understand by using your brain. 1% of the concept to be remembered and 99% of the concept to be, friends, understood. So more briefly you understand the concept, more easily you can answer the questions. Okay, now let's continue. The splicing. What is the speciality of this particular splicing? You all commonly know that we will cleave the introns, which are non-coding parts. We will join the exons, which are the coding parts. We all commonly know that. So, friends, you know one interesting line about the splicing. The interesting line about the splicing is its accuracy. What, my friends? It's a great accuracy. Always listen my words very carefully. Friends, splicing, you know, right, we use it to call DNA replication is the most accurate process. Friends, why does the DNA replication is the most accurate process? I use it to say because the reason behind that, our DNA polymerase enzyme will make only one mistake, just one mistake out of 10 power 6 nucleotides added. Just one mistake, that's all. But my friends, all the students pay attention. This particular splicing, always remember, it never ever cut even a small part of the exon. Because, friends, you all know, right, exon is what, friends? Exon is the coding region. If we cut the coding region, protein synthesis cannot occur, right? But friends, the speciality of the splicing machinery is that they never ever cut even a small part of the exon also. That's why we can say splicing is the most accurate process in the cell after the DNA replication. So friends, for the period of 10 minutes, please listen to me very carefully. Splicing is very confused if you don't understand. So I'm requesting all of you friends, pay utmost attention. It is very confused friends. If you don't understand in a correct manner, look into this. This is the 5 prime exon, and my friends, this is the 3 prime exon. Always listen. If the splicing wants to occur, what we should do, listen to me carefully. We have to add the 5 prime exon and we have to add the 3 prime exon together. Very simple point it is. Now friends, where exactly we have to cleave, that is also an important point. The cleaving always occur at the splice site. What my friends, all of you remember to me carefully, where this particular splice site is present. See, the site where we cleave, we can call splice site. Since this splice site is located towards the 5 prime direction, we can call 5 prime splice site. And this splice site is located towards the 3 prime direction, we can call it as what friends? 3 prime splice site. Kindly don't confuse my friends. And and what is the point I want you to remember? Splice site contain phosphate group. Don't confuse with this. Everywhere splice site contain what group friends? Phosphate group. And friends, the entire splicing, what friends, the entire process of splicing exclusively depends on a particular point we call, my friends, branch point. What is this particular branch point is actually made up of? Friends, this particular branch point is generally made up of adenine. Don't forget this. Now you only look into this. They have written here the 5 prime site. This is the 5 prime splice site, right friends? Yes. Near the 5 prime splice site, which sequence is present friends? GU sequence is present. Correct na? Yes. And at the 3 prime splice site, which region is present? AG region is present. This is the branch site. Branch point contain adenine. Poly Y sequences are present after the branch point. That is called pyrimidine rich region. Now you can ask me, sir, are these common sequences? Same. I can tell you these are all the generalized sequences. So you should not think that every time GU sequence, AG sequence and pyrimidine rich sequences are present on the mRNA. No, I never say that. They are generalized friends, so mostly they only present, but not all the time. But this is common. Every mRNA contain what site, my friends? Branch site, which is extremely important. Okay, friends, every one of you pay attention, please. Now, splicing mechanism. First, listen to me carefully. First, I am explaining to you in this particular slide how does the splicing occur. I am explaining you just how does the splicing occur. In the next slide, we will understand different type of splicings, named as simple splicing, spliceosomal mediated splicing, alternative splicing, trans splicing. Many type of splicings are there. In this particular slide, we are understanding how exactly splicing occur. I mean to say how the introns are exactly cleaved and how the exons are joined together. That is what I am going to explain all of you in this particular slide. Kindly don't confuse with this point. All of you pay attention, friends. This is what 5 prime exon and this is 3 prime exon. I will explain twice. Pay utmost attention. And this is the intron and this is also intron. And friends, all of you can see the clear picture. This is what friends? Branch point. What did I said to all of you friends? The branch point contain what friends? Branch point contain adenine and adenine having OH group. What group? OH group. You know, right? What what oxygen is doing here, my friends? Here oxygen acting as a nucleophile. What, my friends? Here oxygen acting as a powerful nucleophile, so which is involved in cleaving the phosphate group. Every student pay attention. Now this adenine, which is having the OH group, this oxygen nucleophilically, listen to me, oxygen nucleophilically come and cut the phosphate group. It will come and cut the phosphate group present at the 5 prime splice site. My friends, half intron came out. See here, half intron came out. Now friends, you know where this particular OH group is added right now? It will go and add near the 5 prime splice site. Once again, my friends, this particular OH where oxygen nucleophilically come and cut this particular phosphate group. Friends, the moment it will come and cut the phosphate group present at the 3 prime splice site, you can see entire lariat came out. And friends, both exons are joined together with the help of an enzyme ligase, and they can form the seal, they can form the nick. I am explaining to all of you one more time. Please pay utmost attention. I know most of the people would get confusion here. I am revising once again. Pay utmost attention, friends. This is the branch point adenine. Okay, this is 5 prime splice site, and friends, this is the 3 prime splice site. Okay, very simple point. These are the introns. Friends, pay utmost attention, friends. Listen to me very carefully. This is branch point adenine. Adenine having OH group, where oxygen acting as what, friends? Oxygen is acting as the nucleophile. Very simple point it is. My friends, this particular oxygen nucleophilically, what the oxygen is doing nucleophilically, it will come and cut this particular phosphate group. Listen to me carefully, friends. This oxygen will come and cut this particular phosphate group. Whenever it cut the phosphate group, half intron comes out. Now this OH group you all able to see here, it added here. Now it will do the second transesterification. This particular cleavage, my friends, we can call transesterification mechanism. One is the first transesterification. This is the second transesterification mechanism. Now here, this particular oxygen will go and cut the phosphate group present at the 3 prime splice site. My friends, now the entire intron came out. 2 exons are joined. Now you know, right, this is the intron that came out. Friends, you know, what do you call this particular intron, friends? We call it this particular intron as the lariat intron. Now you can ask me, sir, why do we call it as the lariat intron? The reason we are calling it as the lariat intron, there is no scientific reason at all. You know, right, friends, farmers used to hold the cow neck with the help of a rope like structure like this. Exactly, it is exactly looking like this, right? They'll make a rope like this, and this area they will, with the help of this area, they'll hold the neck of the cow. So friends, whenever the intron comes out, intron also look like that only. That is the reason, my friends, we just given the name lariat intron. That's all. There is no scientific reason behind that. So friends, what is the point you people has to remember? Whatever the type of splicing it may be, it doesn't matter. The mechanism is same. What do you call it, that particular mechanism, friends? We will be calling that particular mechanism as the transesterification mechanism. What is that, friends? Transesterification mechanism. Whatever the type it may be, whatever the type of splicing, mechanism is common. That is, with the help of what, friends? Branch point, we are cleaving that particular splice site. Now let's talk about the type of splicing. We understand what is the generalized mechanism of the splicing. What do you call the mechanism? Transesterification. Now friends, what are the various type of splicings? The first type of splicing, friends, we will be calling it as the self-splicing. You know friends, why I have used a word self-splicing. The reason I am using a word self-splicing, it did not require any external protein help. It did not require any external enzyme help. With the help of branch point only, introns can be cleaved out in a very simple manner. That's why we are calling it as the self-splicing. But all the time, self-splicing is not possible. That time, you know what the mRNA will do. mRNA will take the help of certain external proteins. What do you call that particular external protein complex? We will understand. We can call spliceosomes. So that's why that kind of splicing we can call it as a spliceosomal mediated splicing. What do you call, friends? We will be calling it as the spliceosomal mediated splicing, where there is a requirement of external enzyme protein complex. And the third one, friends, we will be calling this as alternative splicing. We will understand everything briefly, friends. What do you call the alternative splicing? Then the fourth one, we will be calling it as, friends, trans splicing. What do you call it, the fourth one? Trans splicing. Four type of splicings are very important. We have to read. Okay, we don't know from which kind of splicing we may expect the question. Friends, there is a great chance we can expect the question from this splicing. They can ask from any kind of a mechanism. Okay, let's talk about the splicing first. Okay, self-splicing. And I told you very clearly, what do you mean by the word self-splicing? There is a no requirement of an external protein. There is a no requirement of an external enzyme. That time only we can call self-splicing. So all the students, please listen to me carefully. There are two type of introns are present. One we called, friends, group 1 introns. Another one we called, friends, group 2 introns. Don't forget the statement at all. Group 1 introns means, listen to me carefully. Anyway, one point is very common. Group 1 and group 2 introns are following what kind of mechanism? Self-splicing mechanism only. Group 1 introns are majorly present in the mitochondria as well as the chloroplast. Group 2 introns, they are also present in the mitochondria, they are present in the chloroplast, and friends, even they are present in the nucleus also. Right? Which one? Group 2 introns. Now every student don't get confused and just listen my statements very carefully. If you understand it, you will get an idea. What is the speciality of the group 1 introns is that group 1 introns do not have branch point A. So if there is a no branch point A, there is a no OH group, then there will be a no nucleophile at all. Then how does the process will occur? That is the only disadvantage with the group 1 introns. I want every one of you has to remember, friends, the OH group can be exclusively donated by the guanine. Exclusively donated by the guanine. Guanine is the one which will donate this particular OH group actually. So all of you friends, all of you look into the picture. That's the only difference, friends. Remaining mechanism is extremely similar. You only look into this. The guanine just add the OH group. That's all. There is no difference at all. Now once the guanine donate this particular OH group, immediately what is going to happen, friends? Once the guanine donate the OH group, that oxygen nucleophilically cut the phosphate group present at the 5' splice site. Now half intron came out. Now this OH group added at the 5' splice site. Now it will go and cut the phosphate group present at the 3' splice site. As a result, what happened, my friends? The entire intron came out and the two exons are joined together by a phosphodiester bond. Simple. By the enzyme ligase. My friends, what is the unique statement you people has to remember? Just listen to me. Group 1 introns are those type of introns which are present in the chloroplast and mitochondria. In case of a group 1 introns, the branch point adenine is absent. So but if they want to show the nucleophilic attack, compulsory it require what group, friends? OH group. Now who is going to donate this particular OH group? This OH group is exclusively donated by the guanine. So guanine externally donate the OH group, then that OH group can act as a nucleophile. It cut the 5' splice site and 3' splice site. Simple point. When you look into the group 2 intron, group 2 introns are normal. There is no problem at all. Group 2 introns are present in the chloroplast, present in the mitochondria, present in the nucleus. Here there is no problem at all. What? They generally contain internal adenine residue as their initial attacking nucleophile instead of external G to form a lariate intermediate. What, my friends? Group 2 intron do not require any external G which will donate the OH group. It's not required. Group 2 intron generally having adenine like this, my friends. You can see, right, the same mechanism, no change at all. In case of group 2 adenine is present, then everything will occur commonly. So I am summarizing. In the group 1 intron, OH group is donated by the guanine. In case of a group 2 intron, no need to donate this particular OH group. Already adenine is present. Now let me summarize one common point. Whether it is a group 1 intron, whether it is a group 2 intron, doesn't matter, friends. Both are doing the cleavage with the help of branch point, that is with the help of the OH group. Do you think either group 1 or group 2 introns, are they using any external protein? Group 1 or group 2 intron, are they using any external enzyme? Not at all. They are not using any external protein. They are not using any external enzyme at all. That's the reason, my friends, we will be calling them as the self-splicing. Please kindly don't forget the statement at all. Okay. Friends, clear everyone? Remaining thing we will discuss in the next class. So friends, maximum 7 days. I think today is 21st, right? By 29th, maximum by 28th or 29th, both unit 3 will be completed, unit 6 also will be completed. So later we will write one test on both units and we will move on to the another 3 units. In the month of April, I will mostly cover 3 to 4 units. Minimum 3 units I will do very full-fledged manner. Okay. Is that clear, friends, everyone? Till the group 2 introns. Remaining thing we will discuss in the next class. Okay. So friends, in the tomorrow morning class, we will start studying about the plant hormones. Oh, that's really great. So friends, please attend every day regularly, friends. Don't miss the flow. You know, attending 1 or 2 days or 3 days is not important. We require a great consistency, friends. The more consistency we have, we reach our success very closely. Okay. So I want every one of you to attend very regularly. Okay, no problem. I'll see you in the tomorrow morning class. Thank you.